good morning, everyone. So I'm Dan. Thank you for inviting me. I'm the opening talk, as you already realized. So let's start. I'll go back and forth. I'm sorry. Yeah. So, all right. So we've been with our mitochondria for the past 2 billion years. We've been with our mitochondria for the past 2 billion years. With the mitochondria for the past 2 billion years. So you probably realize that it was once a free-living bacterium, and it lost its independence, because of this doesn't work. No, doesn't work. Sorry. Yeah, now it works. So the loss of independence was partially because of the loss of genetic material. So the genetic material was either lost totally or was transferred to the host genome. And this created a bigenomic system. And the bigenomic system was accompanied, as I said, by loss of independence. It means that the mitochondria cannot be grown outside of the cell, and the cell cannot be grown without their mitochondria. So it's not only negative. Loss of independence sounds to me, we cannot live alone. But in the end, you have to co-adapt with your host. And this is exactly what is happening in each and every cell of your trillion cells that you call yourself. So I'm going to talk mostly about the co-regulatory aspect of this collaboration, all right? So why did I mention co-regulation and collaboration, co-evolution, and co-whatever? So in the end, when you come to think of that, the mitochondria has to function. And the bigenomic system is operated in 3 different levels. One would be too many things to push. So, So the first would be the protein-protein interaction level at the oxidative phosphorylation system. 4 out of the 5 oxphos complexes, 1, 3, 4, and 5 complexes, these things, yeah? This 1, 2, 3, 4. So 5, yeah? They are comprised of mitochondrial DNA and nuclear DNA encoded factors. So, and they require direct protein-protein interactions. The second level would be at the mitochondrial ribosome, which is comprised of more than 70 subunits, protein subunits, all encoded by the nucleus, translated in the cytoplasm, imported into the mitochondria, and they have to interact directly with ribosomal RNA, the yellow stuff here. The ribosomal RNAs, which are encoded by the mitochondrial DNA, transcribed by the mitochondrial DNA, and of course the tRNAs that are mostly encoded by the mitochondrial DNA. So protein-RNA interaction. The last level of interaction would be the DNA itself. So the mitochondrial DNA has to be regulated somehow, and all the proteins that regulate the mitochondrial DNA are imported from the nucleus and bind the mitochondrial DNA and regulate its function. So these are 3 levels of interactions. But most of you who are biologists, so yeah, she's not. But she's very nice. So in any case, you're a geologist, is okay. So I'll leave you alone, just a second. So the point is that every function in the cell incorporates interactions. So what is unique about the mitochondria? So at least in animals, it is unique. It is unique because of the 10 times mutation rate of the mitochondrial genome. Well, it accumulates a lot of mutations. And in the end, since these factors have to collaborate, the mitochondrial and the nucleus, we need to co-adapt. And this is going to be heard by most of the people that are going to talk during the day, I'm not going to talk about these mutations. I'm going to talk about co-regulation, because this is almost ignored. Why co-regulation? Well, first, because it is very different. In the beginning of the 1970s, the late Yossi Aloni here and the late Giuseppe Attardi discovered in HeLa cells that the mitochondrial DNA is being transcribed in humans. It is being transcribed in its entirety in a single molecule, which a polycistronic molecule, which in turn is cleaved into individual mRNAs and sometimes dicistrons. So it retained some bacterial identity. Later on, after in the years that came later on, the factors that regulate mitochondrial DNA transcription were isolated, and it is a handful. That's it. In fact, 5. The RNA polymerase of the mitochondria, POL-RMT, transcription factor A, TFAM, transcription factor B, 2, TFB2M, the elongation factor, TEFM, and mTERF, the termination factor. That's it. And they are all encoded in the nucleus, imported into the mitochondria, and work only in the mitochondria. Aha. So there comes the dogma. The dogma states that the mitochondrial DNA transcription is separately, completely separately regulated from that of the nucleus. Because when you come to think of that, nuclear genes are not polycistronic. They are all transcribed alone. They are alone regulated. There are some co-regulation occurring within some biochemical pathways, but it's certainly not at the transcriptional level. So how do these things collaborate? The dogma states that the collaboration is being maintained by signals traveling from the mitochondria to the nucleus, back and forth. That's it. But we thought differently. Not that differently, but in addition, we thought to ourselves, in evolutionary eyes, is it not possible that during the past 2 billion years, can you comprehend that we've been with our own mitochondria as eukaryotes? Has there not been some adaptation of the mitochondrial regulatory system to the host? So at first, we have to ask a very basic question. Do we have any evidence for co-expression of the mitochondria and the nuclear genes in the level of the RNA? This question was asked by Gilad Barshad. He was still, when he was still a PhD student, he recently got a PI position in a university in Israel, in the Technion. Doesn't mean anything to you, but I'm very happy. So Gilad got his position about blah, 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 but in any case, when he analyzed RNA-seq experiments coming from 48 different tissues, he found very strong evidence for co-expression. In fact, what you see here is co-expression of nuclear DNA encoded genes, some more than others. This is heat map for you, those of you who are not very familiar with this thing. So these are nuclear DNA encoded genes. These are mitochondrial DNA encoded genes, but I want to draw your attention to these. Weak, but still very significant and very different from the random. So we have co-expression of mitochondrial DNA and nuclear DNA encoded genes of the oxidative phosphorylation, very different from the random. And Gilad suggested that co-expression may allude to the possible co-regulation. How do you answer this question? You analyze upstream regulatory elements of oxford genes in the body, and this is what he did. He analyzed CHIP-seq signals in upstream regulatory elements of all the oxford genes, and one found preferential binding by a bunch of transcription factors, of which 2 attracted our attention. And these were CTAP, enhanced binding protein the mitochondria, bind the mitochondrial DNA in vivo in sheep qPCR as well, and they bind to negatively selected sites. There is a question where they bind. They bind within genes, not in the usual regulatory element. No, regulatory element. No. Regulatory elements. Oh, yeah. So, too much. Regulatory elements within, not the usual regulatory elements in the mitochondrial DNA, but within genes. But when you come to think of that, the mitochondrial DNA is pretty compact. So it could talk in more than one language, more than just the protein coding genes. The same sequence can also code for regulatory elements. Stimulating, isn't it? I'm not going to talk about it today. I'm going to ask a more basic question. First, if you believe what I showed you right now, it means that co-regulation is very prevalent. If it is prevalent, but it is prevalent across many tissues, but tissues harbor many, many, many cell types. So Hadar Medini dived into one cell type. She chose to go to beta cells, the pancreatic beta cells. And why so? Because insulin secretion and insulin gene expression is highly dependent on the mitochondria. So we reason that since not all beta cells secrete insulin in the same rate and express the insulin gene in the same rate, maybe it is connected in some way to the mitochondria. You know, it's a maybe, but we need to be shown. So she analyzed single cells RNA-seq experiments coming from humans and from mice. And what she saw is a cool finding. What she found is a group of cells that had this yellow stuff. I'm not good with my left hand, I'm sorry. So the yellow cells are highly expressing the mitochondrial DNA genes, and the purple cells are expressing lowly the mitochondrial DNA genes. And these two groups of cells were identified in all the individuals that she analyzed in several independently gathered beta cells from humans. Okay, so the cells are divided according to mitochondrial DNA gene expression. What happens in the nucleus? Well, the same very cells had higher or lower gene expression of the OXPHOS genes. Okay, so we're talking about co-regulation of co-expression, at least, of the mitonuclear genes in two different types of beta cells. Does it mean anything? It appears that the cells that expressed highly the mitonuclear gene expression also had high expression of the insulin pathway, including the insulin. Now the plot thickens. So it means that higher mitonuclear gene expression correlates with the expression of insulin. So what will happen if you perturb it? In fact, you can in type 1 diabetes. And this is exactly what Hadar is doing right now. She analyzes single cells RNA-seq experiments coming from type 1 diabetes. So until now, what I showed you, I hope that you were persuaded that that mitonuclear co-expression is pretty prevalent across tissues, and it could be seen in the single cells level in certain cell types. So what will happen if mitonuclear co-regulation copes with or maybe changing physiological conditions? It should react to that. It should retain the same. So we thought to ourselves, what would be the best system to study? Development. And why development? Because it is a dramatic physiological change. And because if you interfere with mitochondrial function in development, at least in model organisms, you stop development. It is a fetal-lethal phenotype. And in fact, usually it is around the blastocyst stage, just before differentiation. So, okay, it is important. Mitochondria is important for development, but nothing is known about mitonuclear co-regulation during the course of development. So Hadar, a very talented PhD student in the lab, asked, how does mitonuclear co-regulation vary during embryo and postnatal development across evolution? Not only in humans or in mouse, but across evolution. How do you do that? Well, you analyze. You analyze a lot of RNA-seq data, which is collected during subsequent stages, during embryogenesis, and right after birth, and after birth towards puberty, et cetera, et cetera. So this was collected, and she analyzed, and we didn't expect anything. We just analyzed all stages, and we looked for the expression of mitochondrial DNA and nuclear DNA encoded genes, and just looked for the dynamics. And what we found was amazing to me, at least. It doesn't do anything, sorry. You see this black thing, the black vertical line? This is the line that marks the birth, our own birth, okay? So you could see that, I hope that you can see that there is a sharp elevation in gene expression, mitochondrial DNA gene expression, right after birth in 4 different tissues. In the hindbrain, forebrain, kidney, and heart, and maybe also in the liver. And it is occurring in all the mammals tested, and surprisingly, also in the chicken. And chicken, as you all know, don't give birth. They hatch, which is a very different developmental scheme. So how come? To make a long story short, it could be associated with the start of locomotion. It could start with the transition from hypoxia to hypoxic environment to a normoxic environment. In fact, the second is the better explanation. I'm not going to show you that, but it is the better explanation. So this is the mitochondrial DNA encoded genes. What about the nuclear genes? The same. So right after birth, there's an elevation of mitonuclear gene expression in different tissues. And I want to point you not only to these 4, not only to these 4 tissues, but also to the liver. And why the liver? This is a short, shaggy story, but I'm going to tell you about that. You know, we accumulate fat. We accumulate fat in very surprising places across our body. But the chick embryo, when it is not born, but when it hatches, unlike us, who we simply eat milk, we get milk from our mothers, he does not get anything. So he has to eat something. And until he pokes upon the first grain that he finds randomly, it has to consume something, and it consumes fat. Where does the fat accumulate in the chicken? Yes, in the liver. Very fatty liver for us is a disease, but for the chicks, it is life. So I don't know if I have enough time, but I can, if I persuaded you, I don't know, how much time do we have? 4 minutes, so I cannot show you the regulatory factors that I can just, you know, these 2 attracted our attention, but it's a long story. I'm not going to go through that, unfortunately, but I can ask you, okay, so the mitonuclear co-expression is not only prevalent, but it responds to physiological conditions, at least across development and right after birth. So we ask the question whether, if it is such an important thing, what will happen if the mitonuclear co-expression is compromised? Since we don't know which are the best factors to regulate this phenomenon, we have to look for different conditions that may point to such perturbation. So we started, not we, but Noam, very talented PhD student in my lab. He grew cells in 24 hours, 1% oxygen, namely in hypoxia. Hypoxia is a dramatic condition that affects the mitochondria. And he prayed forgot that something will happen to the mitochondria, and his prayers were only partially addressed. So, because, we bow to the results. These are the results. So what he found, that while growing 19 different cell types, I show only some of the results, he sees a consistent reduction in mitonuclear gene expression in response to hypoxia. But both genomes go down. So the co-expression is maintained. Now what? So Havird came with a different idea. The idea is that, 2 minutes, thank you. So it is the end, almost the end. So Havird came with a different idea that we might want to look at diseases. So, I'm sorry to say about things about COVID. So the first thing that came to our mind is COVID-19, not because it is so prevalent, but because Kodo et al. Published a nice paper where they infected cells with various types of viruses, and they noticed that SARS-CoV-2 prefers to replicate in glycolytic environment. So we came with the following hypothesis, that the SARS-CoV-2 infection will create a glycolytic environment not just by upregulating glycolysis, but by interfering with mitochondrial function. And the cell will respond by upregulating glycolysis. Did you notice how much I moved my hands? Yeah. Now we have to show that. So Havird tested 14 different data sets. People sequenced like hell. They sequenced all the samples that they could get from blood, upper respiratory tract, from patients' control. And it is a lung disease. So we expected to see something in the lung, and there was none. But in the blood and in single cell sequencing, we saw that there was a dramatic reduction in mitochondrial DNA gene expression in the patients as compared to controls. So if the mitochondrial DNA goes down, the nuclear will do the same. No. So here comes the perturbation. So in COVID-19, we observed departure for mitonuclear co-expression. And, that's the end, I'm sorry. So you saw elevation, as expected, elevated glycolysis, but also elevation of the Ross scavenging machinery, suggesting that we might be correct. And that's the last slide. Is it happening only in COVID? Well, that's a meta-analysis. So it happens also not only in COVID, but in 4 different types of cancer cells and also in Alzheimer's disease. So it might be clinically important. I think that I already spoke about what I want to convey here. I hope that I persuaded you that mitonuclear co-expression really exists, it is important, and it is regulated. How? It's a big question. This work was done by many people that I mentioned. I like very much to collaborate with Israelis and with foreign collaborators. I cannot do anything without funding. I cannot do anything without the mother of the lab, Sarah Dadon. And I'll be happy to answer any question that you have. Shocked. Go ahead.
Thank you for your talk. It was really interesting.
Cool.
You gave an example where there was dysregulation because of a disease causing a departure from co-regulation. Is it possible that, on the contrary, departure from co-regulation might lead to certain diseases as well in a reversed way?
In fact, this is this is the thing that we wish to understand, because until now we did not find a condition or some chemical that will disturb the mitonuclear co-expression. In this way, we could induce the co-expression, learn more about, interfere with the co-expression, and then learn more about the mechanism that underlies it. It's a good question. Thank you. Yeah.
What is your best guess on what is coordinating co-regulation?
This is my best guess. In fact, we analyzed more than around 80 known regulators of transcription, of mitochondrial transcription, post-transcription, and mitochondrial biogenesis, and two of these factors that turned out to be to have the same co-expression pattern with a mitonuclear right after birth. So PGC1 alpha, and again, CCAT enhancer binding protein beta. So I don't know if it is only transcription, but these are the ones that were best correlated with the expression. I can tell you that we already manipulated the expression of CBP beta, and it does interfere with mitonuclear co-expression. But, you know, I cannot talk about everything now. Thank you so much.
All right, well, thanks for the introduction, and thanks for putting on the symposium. I'm looking forward to hearing more about everybody else's work. But I'm here today to tell you about a lesser-known aspect of mitochondrial bioenergetics, and hopefully convince you of horizontal gene transfer of this alternative oxidase pathway into multiple metazoan lineages from independent donors. So my talk today is going to bring us to the inner mitochondrial membrane, where one of the most important mitochondrial processes takes place of oxidative phosphorylation. So if we think back to when we first learned about how mitochondria made ATP through oxidative phosphorylation, this is a figure we would see in textbooks, right? An electron transport system with two entry points for electrons through either complex 1 or complex 2, but just one exit, right? And even in more detailed views of the electron transport system with multiple entry points through beta oxidation or other processes that converge at the Q junction, from there, the only way out is through what I call the cytochrome pathway, where complex 4 tightly regulates the flow of electrons to reduce molecular oxygen to water. Right? This is how our mitochondria consume oxygen, but it just shows this one exit. And these depictions are often based on mammalian or other vertebrate animal mitochondria, which is not representative of most eukaryotes. So what's missing here is that most eukaryotes have a second exit to their mitochondria, so this alternative oxidase pathway, or AOX. So in taxa that have AOX, they can maintain what I call metabolic flexibility. So in the presence of cytochrome pathway inhibitors, of which there are many ecological and physiologically relevant inhibitors of this pathway, taxa with AOX still maintain the capacity to perform some oxidative phosphorylation, but more importantly, I would say, is maintaining the inner mitochondrial membrane potential through the flow of electrons through the system. And that membrane potential set at the correct level is required for many other processes outside of ATP production that are important for mitochondria. So most folks that I talk to about mitochondria are unfamiliar with the AOX pathway, so I'm going to tell you everything I know about it, starting with the phylogenetic distribution of this across the tree of life. So this is work I've done with Alison McDonald recently, and I'll just share some highlights. So among the prokaryotes, AOX is only found in proteobacterial genomes, not other bacterial lineages and not the archaeans. So that's interesting for other reasons. When we come into the eukaryotes, it's found in every streptophyte, every land plant genome has an AOX in it. That's been known for some time. When we go to fungi and protists, AOX presence is the rule and its absence is the exception. Now, into the metazoans, and this is really this is where we had our main motivation for doing this review here. We've known, thanks to Alison's work about 20 years ago, that AOX indeed is present in metazoan genomes. But now that we have more genomic resources and sequences available on NCBI, we're able to fill in some gaps of where it is and more importantly, where it's not. So we've known that it's been in tunicates, for example, bivalves, most cnidarians, copepods. It's notably absent from model systems like any vertebrate animal lacks AOX. We've known that for some time. One thing that I'll highlight today is that we found evidence of horizontal gene transfer of this alternative oxidase pathway into multiple metazoan lineages. The way I'll tell you how we came to this conclusion, and in part it's due to another thing we discovered, is that AOX can act as a barcode. So it has diagnostic sequence motifs that will get you to the kingdom, phylum, or even lower level of organization. So in order to understand how AOX can act as a barcode, I need to tell you a little bit more about its structure and its function. So it's strictly nuclear encoded. It's a terminal oxidase, similar in function to complex 4, but it requires iron molecules for its catalytic function. And those iron molecules are held in place within the enzyme by strictly conserved glutamic acid residues that are nearby an alanine or a histidine. So in this 3D structure of AOX from a fungus, I've highlighted those in red. And across hundreds and hundreds and hundreds of AOX sequences across the tree of life, these glutamic acid iron binding sites are 100% conserved, which is quite remarkable. But how is that diagnostic? Well, if you look at those iron binding sites that I've shown the 4 here, and I've highlighted those glutamic acids, if you look just upstream or downstream of those iron binding sites, here is where we have the taxonomic signature of different taxa. So here are some examples of that. If we isolate an AOX sequence from a land plant, it always has this motif at iron binding site 1, cramlet. And no other taxa has that at iron binding site 1. Now, I'm not going to try to pronounce these other motifs for protists and fungi, but the similar thing is going on. Those motifs are only present in the protists, for example, and not any other lineage. Okay, so in this way we can get some information just by looking at a couple of amino acids. At these other iron binding sites, it gets a little bit more complicated. But one of the things we wanted to focus on is being able to parse between a metazoan origin, a metazoan AOX, and a non-metazoan AOX. So to facilitate that, we've developed this flow chart that the only information you need on hand is an AOX amino acid sequence, and to identify the amino acids upstream or downstream of those iron binding sites. And this will get you to the phylum level or lower within metazoans and other taxa. Now this may seem quite simplistic and crude even, but guess what? It works. It works 100% of the time, validating this a couple of different ways, determining a metazoan or non-metazoan origin. And this becomes important because when we were looking at trying to find new animal taxa with AOX, we started to find some what we call aberrant AOX sequences. So these are AOX sequences attributed to a metazoan, but they have non-metazoan iron binding site motifs that match other taxa. So we flagged those sequences as aberrant, and then we also used a phylogenetic approach, built a phylogenetic tree based on AOX sequences of a high confidence alignment with species known to have functional AOXs. Then we added in our aberrant animal sequences and see that they cluster outside of the metazoan clades. Right? They cluster with alpha proteobacteria or some fungi or protists, right? But there's an important distinction here. There's 2 processes that can result in the same outcome. The obvious one is it's contamination during isolation of nucleic acids, right? That could be one way we get these aberrant AOX sequences. Another is a real process of horizontal gene transfer of this gene into these lineages. And we've parsed between those using an approach, a gene-based approach of blasting back that aberrant AOX. When we had information about where that was on a contig or a chromosome, look for nearby genes upstream and downstream. If it blasts back to that focal animal, that's consistent with perhaps that being a real thing, and it's been perhaps horizontally transferred. If we find non-animal genes on nearby that AOX, it's likely contamination. So using those methods and some others, we've concluded that there's possibly multiple independent horizontal gene transfers of AOX into metazoan lineages. So within rotifers, within some nematodes, within springtails, this is a subclass Columbola within hexapods, and just one example in an insect, a fungus gnat. I'll just highlight just the springtails here, so I have more data on that I'll show you. We're zooming in here on the AOX tree, and we've got the springtail and fungus gnat AOX sequences forming this monophyletic clade with this protist group, the Oomycotas. And among the potential donors of AOX to springtails and the fungus gnat is Pythium, this Oomycota protist genus, Pythium. Now that is notable because we've actually known that springtails have had lots and lots of genes horizontally transferred from diverse sources like bacteria and fungi, but also Pythium protists. And we've known actually that AOX was one of those genes for some time. It just wasn't obvious because in 2017, when you take this springtail gene and you go into the supplemental material down to line 721, and you see this entry here that's most closely related to a Pythium species and a uniprot ID that was uncharacterized in 2017. Now, if you look up that same accession at uniprot today, you'll see it's now encoded, annotated as an alternative oxidase. So this gives me some confidence that our methods to parse between contamination and horizontal gene transfer might be a good method. These gene-based approaches are all fine, but I really wanted to functionally test the presence of AOX. And the benefit of AOX is it's very straightforward and clear to test for the presence or absence of a functional AOX enzyme. So I started rearing springtails in the lab and used high-resolution respirometry on isolated mitochondria to test for the presence of a functional AOX pathway. I also had some other species as a positive control, a copepod with a known AOX, Tigeropus californicus, and then a negative control as a brine shrimp, a species that does not have AOX. So here on this figure, we've got these different respiratory states that I've induced by adding different substrates to turn on the oxphos machinery, and then the cytochrome pathway inhibitors to shut down the cytochrome pathway, and then shut down the AOX pathway. So you can kind of view this as a time series of sequential events going from left to right. We're measuring oxygen consumption. So let's look at a species without AOX. Any mammalian mitochondria you put in this machine, you're going to get an oxygen trace that looks like this. We give it the substrates that it needs to ramp up oxidative phosphorylation. We get to some level of oxygen consumption. Then we add a cytochrome pathway inhibitor at saturating levels. And what happens? Oxygen consumption goes to zero. What's going on here? We've blocked the only exit for electrons to interact with oxygen, right? So this is a experiment you would see and do in any mammalian mitochondria, a vertebrate animal one. What about species with AOX? Well, in the presence of saturating cytochrome inhibitors, we still have significant amounts of oxygen consumption from isolated mitochondria. This is entirely consistent with the presence of a second exit, the alternative oxidase pathway. We've only blocked one, and we still have this pathway open to consume oxygen, and we're seeing that signature quite clearly. Only when we also add an AOX inhibitor do we get oxygen consumption to stop. So where do the springtails fall out? They have a pattern that's entirely consistent with a functional AOX. So together with our phylogenetic and sequence-based approach and these functional tests, I've convinced myself, at least in this one species of springtail, we've got evidence of horizontal gene transfer of this alternative bioenergetic pathway into a springtail from a protist source, perhaps. There's other springtail species we're working on testing now, and also we need to do these functional tests in these other taxa, but an obvious open question here is why? Why would this potentially horizontally transferred gene be retained? And is it adaptive, or is it just a random drift-based event? So there's a lot of work to do on this, and we're trying to now to come up with some ideas for as why this might have been retained. And what do these taxa that have putatively had this horizontally transferred in, what do they have in common? One thing we're working on now is hypoxia tolerance. All of these taxa tend to live in quite hypoxic environments at certain life stages, perhaps, and that might be one benefit of this alternative oxidase pathways. There's certainly other answers to this question. But if this is something that is interesting to you, I'm looking for PhD students at my newly formed evolutionary bioenergetics lab at Iowa State University. We're working obviously on this alternative oxidase pathway, but we also do work on mitonuclear coevolution and regulation, and starting work on bioenergetics of pathogens as well. So I would look forward to hearing from you all about that. And that's all I've got for you, so thanks. Absolutely, yeah, that's one of the proposed mechanisms of what we know AOX to function in plants, for example. It's known as part of the stress response. So it prevents over-reduction of the electron transport chain, and that's when we start to see a lot of reactive oxygen species production. So it could be, that could be a part of the equation here as well, certainly. Yeah, thanks.
All right. Hi, everyone. So today I would like to talk about mitochondrial DNA copy number variation in songbirds and how it relates to the sperm midpiece length. So to start with, sperm cells are among the most diverse cells in the animal kingdom, and my lab at the Natural History Museum in Oslo have been studying sperm evolution in passerine birds for over 20 years. So passerine sperm is especially interesting because they are very different from non-passerine spermatozoa, so for those who are not familiar, sperm cells are composed of two main components. So we have the head containing the acrosome and the nucleus. Here you can see it, numbers 1 and 2. And then the rest of the cell is the flagellum, which has the midpiece and the tail. So what I wanted to illustrate with this figure is the morphological diversity of sperm cells between relatively closely related taxa. So here we see 3 passerine species, and for example, so the first one is twice as long as the other 2 and has a much larger acrosome, and so the variation is really big across all of these passerine species. So as one of my PhD topics, I am specifically interested in the midpiece, which is the part of the cell that contains mitochondria and generates energy for the movement of the cell. So songbird midpieces are typically formed by several mitochondria fusing together at the end of the spermatogenesis, and then this long mitochondrion wraps around the flagellum. So due to this, we are not able to use the usual transmission electron microscopy to assess the number of mitochondria that form the midpiece. So this brings me to the main question. So do more mitochondria need to fuse together to form a longer midpiece? So in general, it's very little is known about the relationship between mitochondria and the midpiece length in passerines. And to date, there has only been one study in passerine birds investigating this relationship. So in zebra finches, there is an inversion on the Z chromosome, which causes significant intraspecific variation, giving rise to 3 different phenotypes. So they have short, medium, and long midpiece. And a study by Kneath and his colleagues has shown that longer midpieces contain more copies of mitochondrial DNA. So then we were interested in seeing whether we can see the same pattern across a range of different passerine species. And so based on theory, we predict that first, longer midpieces would be formed by more mitochondria, and that we could use mitochondrial DNA copy number as proxy for the number of mitochondria that fuse together to form the midpiece. So to investigate this, we collected sperm samples from 19 passerine species representing 10 families, and we collected them in southern Norway during 3 breeding seasons. And here in the center, you can see, I wanted to showcase the variation in midpiece length across all of these species. So the arrows point at the shortest midpiece, which we see in the bullfinch at 1.72 micrometers, and then the longest is in the reed bunting at 250 micrometers. So we extracted DNA from these sperm samples using 4 individuals per species, and then quantified both mitochondrial and nuclear DNA using digital droplet PCR, and then used the ratio between mitochondrial and single copy nuclear gene as a number of mitochondrial DNA copy per spermatozoa. So we expected to see a sort of linear relationship between the midpiece length and the mitochondrial DNA copy number, but what we saw, in fact, looked more like a U-shaped relationship. So it was very interesting to see that there is quite a lot of individual variation, as is indicated by the error bars, and then, but the overall range was not that big as well, because it goes from, so the species with the lowest mitochondrial DNA copy number is the house sparrow. It has on average 9 copies per spermatozoon, whereas reed bunting, which has the longest midpiece, has 60. So it doesn't vary that much, because we've also seen, like in other taxa, even within the species, the range can be similar, and this is across species here. It was also quite interesting to see that the bullfinch, which I mentioned as having the shortest midpiece, had almost as many copies of mitochondrial DNA as the species with the longest midpiece. So then we cannot be exactly sure why we see this pattern, but there are some points of discussion that I would like to go through. So I've now mentioned the bullfinch a couple of times, but what I didn't say is that in this species, the sperm cell is actually quite different from all the other passerine species. They, as you can see in the photo here below, they do not have this large acrosome, and they look more like immature spermatozoa or spermatids than actually mature passerine sperm cells. So it has been shown that in this species, the final stages of spermatogenesis are skipped. So that results in this neotenous spermatozoa. In addition to that, in other taxa, it has been shown that mitochondrial DNA content undergoes strong reduction at the final stages of spermatogenesis as a pre-fertilization barrier of paternal mitochondrial DNA transmission. And so if we connect these two patterns, we speculate that in passerine birds, mitochondrial DNA reduction also occurs at the last stages of spermatogenesis, and that could explain why, for example, bullfinches have so much mitochondrial DNA compared to other species. But then even if we're saying that we have this U-shaped relationship between the midpiece length and the mitochondrial DNA copy number, the relationship is still quite flat. So we're wondering, maybe there's actually like an optimal range within which passerines can have the mitochondrial DNA copy number without having some negative effects to the cells. Because in human research, it has been shown that sperm cells that have more copies of mitochondria usually are found in infertile men. So there must be some sort of relationship between mitochondrial DNA copy number in sperm and the quality of the sperm. So so to conclude, our study is the first one to demonstrate that mitochondrial DNA copy number in mature spermatozoa does not correlate with midpiece length across passerines. And we also see that the interspecific variation is not so high, and our findings suggest that mitochondrial DNA might be removed at the late stages of spermatogenesis. But of course, we're not entirely sure, and more studies would be necessary to confirm this. So we would need to study to compare immature versus mature spermatozoa and see how mitochondrial DNA copy number changes during the maturation of the sperm cells. So with that, I would thank you for your attention, and thanks to my lab members, and thanks.
I see the variation in, was it copy number that was on the y-axis?
Yeah.
How much variation did you see in the actual midpiece length within species?
So here we just used species average, but it would be good, for example, like to collect more samples and then you can measure per exact individual and see how it is related to that. So we thought about that, but we haven't done that yet. But that would be interesting to see as well.
Great talk. So I know that you focused only on the mitochondrial DNA copy number, but there was a recent paper, not from birds, from humans, that there's a loss of mitochondrial. We talked about the loss of mitochondrial DNA in the maturation of the sperm in human cell, in human sperm cell. And the main thing is about how the mitochondrial function. I mean, these cells have to move and to compete for fertilization. So, if I understand correctly, there is no correlation between the meat piece length and the number of mitochondria. So, what is your hypothesis? What does correlate?
I'm not sure exactly, because we tried looking whether there's any correlation with, say, like the strength of sperm competition or so, but of course we didn't have it for all of these species, so I didn't have the data, and we didn't find any correlation. So always the shape or like the pattern was kind of U-shaped. So we still don't know exactly why we see what we see.
In other words, are you going to gene expression as well?
So we briefly discussed that a bit, that it could be interesting to see and probably would help answer some of these questions. Yeah.
There's some interesting work in birds and mitochondrial and sexual selection. I wonder if you saw any correlation among the species that you looked at, where some have like conspicuous male colors or something, and whether or not that correlates with mitochondrial copy number in sperm.
We haven't looked at that yet, but yeah, it'd be interesting. Yeah, because I feel like, I mean, when we did this, we were also like thinking about, okay, which traits we can, check this against. But yeah, so far.
Sperm competition particularly rampant in birds?
Yeah, and it is quite different across species as well. So like in bullfinches, for example, there's almost no sperm competitions. They're very like monogamous. But then you have other species that there's a lot of multiple copulations and extra pair copulations. So potentially there's some influence, but I don't know for now.
And one final question. Is this graph like corrected for phylogeny, this correlation?
Yeah, so I did PGLS and yeah.
So my question was related. So I mean, it is the same question, but do you say anything about how copy number changes, like on the phylogeny? Like you showed how the piece changes along the phylogeny. Is there phylogenetic signal in the mitochondrial copy number?
We didn't get that, no. But then also like in the way that we sampled now, so we had 10 families, but most of the families just have one representative. So I feel like also we need to like increase the sample size and then check a bit more.
So you mentioned that in bullfinch that it has like they're less structured, like they're less fused, the mitochondria. Have you looked into like how networked the other ones are and whether or not it's influencing your kind of curve?
Sorry, can you repeat that?
Yeah, so have you looked into kind of internal structure, how fused or how punctate the mitochondria are within these, and like there's any kind of relationship there?
Oh, like in other species, you mean?
Yeah, or even
Now I can hear you.
even within this group or within all of these, have you looked at kind of like how fused or how punctate the various mitochondria are within this?
So I think that in all of these species, they're just fused, but like I haven't studied that more in detail.
Okay, cool.
But because I think only the bullfinches are the ones where we can see unfused mitochondria, but then in most of the passerines, they just fuse and then you cannot tell them apart.
Cool, thank you.
Okay, thanks.
All right, thanks very much. So it's been touched on a little bit already, but I'm going to talk about mitonuclear coevolution in eukaryotes. So in our lab, we think about a lot of big questions in evolutionary biology. We have people at this conference giving talks on speciation, environmental adaptation, aging, all sorts of fun stuff. But this is a mitochondrial symposium, so we know that all these things are in fact driven by mitochondria. And maybe only mitochondria, according to some people. But I'm going to talk about some molecular evolution work that we've done that sort of has big implications for all these ideas. So again, most people in this room probably know that in almost every eukaryotic cell we have at least 2 genomes, a big one encoded by the nucleus that most people pay attention to, and then a smaller one, maybe in multiple copies, encoded by mitochondrial DNA that is near and dear to all of our hearts. And if we look inside a mitochondria, we see all these little molecular machines that are made up of pieces and parts that are encoded by both of these genomes, right? And the electron transport chain, where we get all our energy, is a great example of this. It's composed of these multi-subunit enzyme complexes, the OXPHOS complexes, that are made up of proteins encoded by both the mito and the nuclear genome. And these things have to come together and function in a very precise, intimate way in order for us to make energy. So this has led to this idea of mitonuclear coevolution, which is very simply that changes in one genome select for complementary or co-functional changes in the other genome. So what this means is that if you have a set of 2 genomes that are paying attention to each other and evolving in response to each other, that lineage is going to have a higher fitness than a lineage where both genomes are evolving kind of willy-nilly by themselves. So there's a lot of evidence for this. I'm going to be talking about molecular evolution studies that have sort of backed this up. I first got interested in this by looking at this plant genus called Silene, which is really interesting because it has some species with slow evolving mitochondrial genomes and then closely related species with fast evolving mitochondrial genomes. If we look at this on a phylogeny of mitochondrial DNA, we can see this where we have these blue species that have these really long branches, and then these orange slow species with super small branches indicating like almost no molecular changes in those. And under mitonuclear coevolution, we would expect that when things go really fast in the mitochondrial genome, they should also go really fast in the nuclear genome. So we started looking at nuclear rates of evolution in these fast versus slow species. And in fact, we did see really fast rates of nuclear evolution in the species that had fast rates of mitochondrial DNA evolution. But we only saw this for a subset of genes called the NMT genes. So these are nuclear encoded genes that co-function and co-localize to the mitochondrial genome. So these yellow OXPHOS genes up here encoded by the nucleus are a great example of these NMT genes. If we look at a random set of genes across the nuclear genome, we don't really see this coordinated increased rate of evolution in those fast species. So that's really interesting, but besides me and the sex chromosome people, nobody really cares about Silene. So we wanted to see whether or not we could apply this to some really broad swaths of phylogenetic diversity. So if we zoom out a bit and look at sort of a more ancient clade, like mammals, we also see quite a bit of variability in mitochondrial evolutionary rates. But this variability isn't dichotomous. There's not a group of fast and slow mammals. It's more or less continuous. So we have species with fast, slow, and everything in between. So under mitonuclear coevolution, we would again expect these rates to kind of co-vary. We would expect there to be this positive relationship between mito and nuclear evolutionary rates if we looked across mammals, for example. And this is called an evolutionary rate covariation, or ERC analysis. And I think this is a really powerful tool to detect mitonuclear coevolution signatures. So I'm going to be showing you a lot of graphs that kind of look like this. But under mitonuclear coevolution, again, we would only expect these rates to covary in those NMT genes. If we look at a set of control nuclear encoded genes, we would expect no real relationship or maybe a much less obvious relationship. So this kind of pattern is what we would expect under mitonuclear coevolution. There might be other things that could produce this pattern as well, but under mitonuclear coevolution, we would expect this pattern. So Ryan Weaver, who's now at Iowa State and spoke earlier about alternative oxidase, did this very exact analysis that I've been kind of setting up. And just to cut to the punchline, he in fact did see this really strong correlation in NMT genes, including OXPHOS genes, and then a lack of a correlation in a control set of genes, the glycolysis genes. So again, this is what we would expect under mitonuclear coevolution. So now we think maybe mitonuclear coevolution is important, not in Silene, this weird genus of plants, but across maybe all mammals, which maybe more people care about. We weren't the first group to sort of take this approach. I think that honor goes to John Warren's lab, where they did a very similar thing in insects, and they found a very similar pattern. Again, tight correlation in NMT genes, much weaker correlation in control genes, suggesting mitonuclear coevolution is also very important in insects. So again, I think this is a powerful tool, and we're interested in applying this across lots of different groups in the eukaryotes to see where mitonuclear coevolution has the strongest signatures, and maybe more interestingly, where we don't see mitonuclear coevolution, and what can we learn about these processes for those exceptions. So one project that we've started very recently is led by Joseph Doobie, a postdoc in the lab, as well as in collaboration with Evan Forsyth at Oregon State University. And this is doing an ERC type analysis in birds. And the cool thing about birds is there's this previous hypothesis in the literature that says that flighted birds, like the albatross, should require really high mitochondrial efficiency and sort of tight co-regulation and coevolution between their mito and nuclear genomes. Whereas if we look at this poor evolutionary miscast here, the kiwi, maybe that and other flightless birds don't need as tight coevolution. So we would expect to see sort of a really strong mitonuclear ERC pattern in flighted birds and a less strong pattern, or maybe no pattern, in flightless birds. So unfortunately, I don't have that data to show you today. I have this data, which doesn't look like the data that I've been showing you before. But this is using this tool called ERCnet that Evan developed, and it basically looks at all pairwise ERC values for all genes across the genome, including mito and nuclear genes. And if you squint your eyes, you might see that the distribution of ERC values in the OXPHOS genes might be a little better than in just random genes across the genome. But this is very preliminary data, so I'm not going to even really comment on it further than that, just to say that if you're interested in birds, keep an eye out for this work at some point in the future. But again, we're interested in these cases where mitonuclear coevolution might not be important at all, or it might not be as important as we think it is. And this got us back to thinking about another study that we had helped with in bivalves. And this was led by Fabrizio Giselli's group, who many of us know. And basically, we found a very similar pattern in bivalves, where we have this tight co-evolution signal in the NMT genes, less of a signal in control genes. But the cool thing about bivalves is they have this, some of them anyway, some species have this weird pattern of mitochondrial inheritance. So in most animals, you get all your mitochondria from your mom. So this is uniparental maternal inheritance, or strict maternal inheritance. But in a subset of bivalve species, they have something called doubly uniparental inheritance. And in this system, if you're female, then you get all your mitochondria from mom, just like normal. If you're a male, then you get most of your mitochondria from mom, but you get some mitochondria from dad, including all the mitochondria and mitochondrial genomes that end up in your gonads and your sperm. So what this does in these DUI bivalves is it sets up these two sex-specific mitochondrial genomes, and these things can be very divergent from each other. And we thought this was really interesting for mitonuclear coevolution, because now we have a single nuclear genome that has to co-evolve with two mitochondrial genomes. And is it co-evolving with them to the same extent? Well, we had a pretty strong hypothesis going in, because ourselves and others have shown that mitochondrial DNA in the male mitochondria is likely evolving under pretty relaxed selection compared to the female DNA. This is probably because female mitochondrial DNA has to function everywhere. The male mitochondrial DNA only has to function in sperm. You can even see that on this phylogeny, where the male branches in blue here are much longer than the female branches in orange in some of these DUI clades. So we thought we would see a strong signature of mitonuclear coevolution with female DNA, less of a strong signature with male DNA. And this work was led by Chase and Raquel, two folks who just left the lab. And this is just coming out in Evolution now, so if you're excited about it, check it out. So we did this ERC analysis with the female mitochondrial DNA, and there it is. We saw, again, sort of a very tight signature with the interacting genes, much weaker signature with the control genes. But what was interesting was that we saw this very same signature with the male mitochondrial DNA. Again, this tight coevolution signature with the OXPHOS genes and a much weaker coevolution signature with a set of control genes. So what this suggests is that even though male mitochondrial DNA is evolving under relatively relaxed selection, there's still really strong selection for it to co-evolve with nuclear DNA. So maybe mitonuclear coevolution is a rule for eukaryotes, right? This was like the one example that I thought was going to be the exception that proved the rule, but it turned out to be like the rule that proved the rule. So I think this is still a very open question, and we need to really look at protists, those unicellular eukaryotes, which represent the vast phylogenetic diversity of eukaryotes, but really haven't been looked at in terms of mitonuclear coevolution. You might be asking yourself, why is this important? And one of the reasons it's important is that if we have these co-evolving sets of genomes within a lineage, then that can create reproductive isolation between lineages. So things that have co-evolved with each other don't play well with things that they haven't co-evolved with. So this is work we've been doing with Molly Schumer's lab. We've identified some mitonuclear genetic incompatibilities in a pair of swordtail species, and even though it's not official, NSF has recommended us to get some funding to extend this across swordtails. So if you're interested in this, please hit me up. And with that, I want to thank our funding sources, NIH and NSF, as well as the lab, who are speaking on all sorts of cool mitochondrial things, mutation rates, aging, selfishness, speciation and adaptation, all sorts of stuff. So please hit them up later on in the conference. And with that, maybe I have time for a question or two. Yeah.
So are you seeing stronger correlation with particular nuclear subunits or ones that are, involved in like activation site or anything like that?
Yeah, that's a good point. And in the studies where we've done that kind of detail, we do see somewhat of a signature between the nuclear encoded proteins that are actually physically touching mitochondrial encoded proteins. It's not quite statistically better or stronger, but the signal is definitely there, and I think it's real. I think it's just a sample size issue. So yeah, there are some issues or some things that are showing the strongest coevolutionary signatures, and some things that are still strong but weaker.
Do you have any sense of the conformational fit to each other, or is that?
Yeah, so in mammals we have really good structures of these OXPHOS proteins, which is one of the reasons why Ryan decided to do that in mammals. So we can say, in fact, in the swordtail stuff, we not only know that there are these genetic incompatibilities like in these genes, but they're at physically interacting sites, which is pretty cool. Yeah.
So, in one of your papers, you talked about the hierarchical selection from mitochondrial genomes to organic.
Okay.
So one of the papers. So does that contribute to the mitonuclear
Yeah, so I think what you're talking about is that, in mitochondrial genomes, they interact at these different levels of selection, right? The organismal level where you have high individual fitness, but then also at the level of the mitochondrion or the mitochondrial genome itself. and those things can often be at odds with each other. So mitonuclear coevolution, as I've presented it here, is all about like individual fitness and organismal fitness. But there might be selection on mitochondrial genomes themselves to be selfish, which would maybe not include coevolution with the nuclear genome. So if you're interested in that, you should go see Joseph's talk on Tuesday. He's sitting right next to you there. But he's going to be showing some cool data on that as well. All right, thank you.
Hello everyone, I'm Finley. I'm a second year PhD student with Flo Camus and Aiden Pierce in London. And the majority of my PhD is on mitonuclear interactions, but today I'm not talking about that. Today I'm talking about mitochondria, about climate in humans. So this was a little side project, but hopefully it'll be interesting. So this is a symposium on mitochondria. There are people here who know a lot more about mitochondria than me, but so that everyone's on the same page, I thought I'd start with the short version of why mitochondria are important. Well, as you may know, they are the powerhouses of the cell. It's commonly said. Yes, thank you. But they also do so much more. They're involved in the tricarboxylic acid cycle, which is metabolism, so they're intricately related to diet. They have a key role in oxidative phosphorylation and ATP synthesis, as well as things like thermogenesis, so regulating body temperature, cell signaling, and apoptosis. So they really are really crucial to life. However, they're also interesting because they have their own DNA. In humans, this is very, very small. It's about 16.6 kilobytes. If you're interested in why the genomes are so small, I'd recommend you stick around for Aiden's talk, who's at 11:45. They have 37 highly conserved loci. So we have 13 protein coding genes. So we have, the genes are in purple, blue on the little diagram. We have 2 RNA genes, which are in green, or light green, and we have tRNAs, which are in dark green. So we also have very, very few intergenic regions, which is interesting. And because they have very small, highly conserved genomes with very small intergenic regions, kind of led to this traditional dogma of if you have a mutation, it's likely to be in a highly conserved region. Therefore, it's likely to be deleterious. Therefore, you'll have reduced fitness. Purifying selection will act to remove these mutations, and therefore, you'll be left with mutations that are selectively neutral. This is the traditional dogma. But this overlooks one thing, which is, what about positive selection? Normally overlooked, but has the potential to be quite informative. And at this point, I have to say, I'm not the person to think about this. There's been a lot of work done on this before, including by some members of the audience, I think, if they're here. But what's changed? So this work was done in the early 2000s, and you may be aware, we now have many, many more genomes and newer methods. So I'm racing through. Okay. For this project, I downloaded 64,000 mitochondrial genomes of NCBI. This is every single publicly available genome that I could get my hands on. It's very similar to the number in Mitomap. So if you've used Mitomap, it's the same thing. Of these, about 25,000 were usable because I need location data. And broader problem, most of them don't have it. Big pain. So following on, I used about 19,000 of these. So this is downsampling. So I have not too many from some countries, which have good location data, so on. This covers about 80 countries. There's a map in the next slide. And I have about 442 SNPs that I can look at. Of these 442 SNPs, lots of them have very, very low allele frequencies, so I'm testing 209. This little thing on the right, over here, is a PCA. This is broadly similar to what you'd see in a nuclear population genetics graph, and this is what we use to account for ancestry. Moving on. We also, over here, have a surplus of environmental data. So some of you may have used WorldClim, which is satellite data from 1970 to 2020. And there's also packages now like ParsClim, which I'd recommend, which give you access to reconstructed paleoclimate variables. So for our data set, we use average values over the last 20,000 years for every country in the world. I'm not going to talk about this in much detail in the interest of time, but if you have any questions about it, please come speak to me later. We also have latitude, which is standard. So we have the genetic data, we have the environmental data, we compare them up together. And what we use is we combine all this data and we apply it in generalized linear models, specifically generalized linear mixed models. And we ask this question: are climate variables significant predictors of mitochondrial genotype distributions? Can we predict where we see a genotype based on climate? So how do we do this? The first thing we need is a null model. This is what you would call the traditional dogma. And this says that the majority of the variation you see will be explained by shared ancestry. Closer related individuals will more likely share the same genotype. So we have to account for this in our model. So the way we do this is we use the principal components from the first slide. You could use the whole tree, but we have 20,000 individuals. Principal components make life easier. But in theory, the information we contain should be the same. And in our null model, we say that the distribution of the genotype that we see is explained by this shared ancestry. The alternative is that the shared ancestry we see is explained by, the genotype distribution we see is explained by the shared ancestry, but also some climate factor. So to start with, we use latitude and annual precipitation as our proxies for climate. The reason we use latitude is because it correlates very, very highly with lots of different temperature variables, and it's very complicated to test all the different variables separately. Annual precipitation doesn't correlate with latitude as well, so we add it in as an extra factor. And what we ask is, does the alternative model better explain the distribution than the null? So what we see, this black line here is the Bonferroni corrected 1% threshold. Everything above the line is better explained by that second model. So we're saying, for all these sites, the distribution of genotypes is better explained by a model that accounts for shared ancestry, but also has climate as a factor. So there are two key takeaways from this bit. The first thing is that principal components are a strong predictor in every site we test. The reason this is important is because it suggests that our measure of shared ancestry is accurate. And the second thing is that we have 33 sites which are significantly better explained by the alternative model. Now, a problem with this could be that you have effectively a single locus, and therefore are you just detecting the same thing and everything's in linkage. So you can see here, purple is highly correlated, grey is not correlated. So you can see for the majority of sites, we have pretty independent regions. If we look a bit more closely, we can see, if we look here, here, and here, we have some sites that are in high linkage. These are all the sites within the control region, or the membrane attachment site. So within this region, we can't really work out what's going on, but in the middle, we largely have good results. We can also see that the hits are distributed across the whole genome. So where are they exactly? If we look at this one, this is a distribution. This is a plot showing where the hits are. The height of the bar is the percentage of hits for a loci that's significant. So of all the SNPs we have in a region, how many of them are significant? The number is the absolute value. So for example, on the right, we can see 100% of hits in tRNA glycine are significant, but there's only one, and we only have one SNP. So that's hence the bar. So there are a couple of interesting things. The first of which is, again, the hits are distributed across the genome. The second of which is complex 1 actually doesn't really stand out. And I think one reason this is interesting is because complex 1 is often talked about, but this result would suggest the reason it gets a lot of hits is because it's big, right? Not because there's anything special going on. We also, if we look at these green bars on the right here, we have some hits in the RNA and some hits in the tRNA. And I thought this was quite interesting, so how many do I have left? In the last bit of this talk, I'm just going to do a little deep dive into these regions. So there's one position here, which is position 3010. This is in the ribosomal RNA, 16S. We have 15924. And this is one of the hits we see in tRNA thymine. Thymine? I think. So this is a plot of both of them. For both of them, there are higher frequencies in Europeans and some Asian populations. And when we do our test of paleoclimate variables, for both SNPs, the temperature of the wettest quarter is the most significant predictor of the ones we test. So this is interesting because it might suggest that it's more of a thermogenesis role. So cold and wet, you have to work harder to thermoregulate your body temperature, which is interesting. That's good. We can see this is work not done by myself, but just collected from the literature, that both of these SNPs also induce structural changes. So at the top here, this is the 3D structure, and this is another one. So you can see this is where the mutation is. It's quite a sharp difference in the ribosomal structure. This one over here is from tRNA scan. This is the site of the mutation. So this is in the anticodon stem, which is important for structure. I haven't worked out how to establish the 3D structure. If anyone has any ideas, please come see me. So both mutations have also been associated with different pathologies. So this has been shown before. So in a nuclear gene, TRMP8, we see increased levels in Europe, but also it's associated with migraine risk. So here we have a very similar thing. We have the classic mitochondrial pathologies, Leber's, cardiomyopathy, myopathy, migraines. And this is kind of interesting because it raises this question of, is there a trade-off between thermogenesis and disease? This is quite interesting. So, rounding off, this brings me back to my original question. Has the human mitochondrial genome evolved under environmentally mediated natural selection? Point number one is that we do have evidence for climate by mitochondrial DNA interactions, or associations at the very least. Adaptation to cold and wet is more commonly detected. That doesn't mean it's more prevalent, it just means it's easier to detect. So this is interesting. And the third one is that when we talk about the mitochondrial genome, we mean the genome. We don't just mean protein coding regions. So regulatory elements could be very, very important, relating to copy number and so on. And with that, I've sped through, but I'd like to say thank you to Flo. She's talking later on today. Ida back in London, the whole lab group. Stefano is talking on Tuesday, and everyone who published DNA with mitochondria, specifically those who included location data, because that was very helpful. Happy to take any questions.
It's very nice talking. Got these statistical relationships, but I wonder if you thought about mapping those SNP frequencies back geographically to see if you have sort of independent origins or selection independently.
Yes, so I've done that. I don't have the plots here because I have 30 SNPs go through. I haven't done it for all of them, but for at least the strongest hits, I have independent origins. So you have repeat mutations, which is what's driving it. Which is quite good. Any questions? So I haven't mentioned it here, but most of my work is on, well, I'd like it to be on mitonuclear interactions. And the reason we were interested in climate is because it's everywhere. And the idea is that mitonuclear interactions can be expressed or exacerbated by stressful circumstances. So the whole point of this was to work out where is the stressful circumstance for mitochondria. So we're doing some work in human populations. So this would suggest, well, give us a kind of insight into which regions might be most interesting to explore. Thank you. Yeah. Mm-hmm. Yeah. Yeah, so it's a good point that Arctic conditions could be stressful, but also it doesn't necessarily work that way because if you in highly tropical environments, you might expect a different diet. So some of the hits, which I haven't talked about, are not in climate regions, they're in kind of Papua New Guinea and those regions. And we're not quite sure what's going on, but it could just be, could be diet, it could be temperature. So there's lots of different things that we've kind of looked at. I haven't specifically looked at climate, like Arctic populations specifically. I've just looked more broadly, I guess. So I could do that in the future. Okay, cool. Thank you very much.
All right, thank you, Sophie and Andres, for giving me this opportunity to talk about one of the projects that I have developed in the last couple of years, and it's still in early development, but I'm excited to share some of the data with you and hopefully chat after my talk. And thanks to Finley, I don't have to talk so much about why mitochondria is important, but I think we can all appreciate that mitochondrial function is maintained by very intimate interactions between the mitochondrial genome and the nuclear genome. So we can envision that any genetic variation in either genome should lead the direct and indirect interactions between these mitonuclear interactions and should lead to phenotypic variation among individuals in the population. So I'm not going to convince you that mitochondria is important. I am trying to convince you that C. elegans is a wonderful model for studying intraspecific mitonuclein studies. Well, C. elegans is a powerful model, period. We have, sorry, not, I don't have. C. elegans have very short generation cycle and can be cryopreserved to avoid accumulation of unwanted mutations. And we can easily manipulate their genome by introducing a single gene mutation to study cellular and organismal phenotypes. And C. elegans is one of the main model to study experimental evolution with large panels of mutation accumulation lines and recombinant inbred line that allow us to map very complex trait with high mapping resolution. But the most fascinating aspect of C. elegans for me is their predominantly selfing lifestyle. Selfing species like C. elegans is greatly affected by drift because of reduced gene flow, but selfing quickly exposed beneficial mutation for selection. So this suggests that in local population around the world, to maintain mitochondrial function, evolution should fix for co-adapted mitonuclear alleles in these differently isolated population. In fact, C. elegans has been isolated across the globe in a diverse variety of environment as naturally inbred lines. This collective effort has resulted in an enormous archive of wild strains that are available for studying natural variation and now are available to the public via the Xenoritis Natural Diversity Resource, and not only just for C. elegans, but for other selfing species such as Brixie and Tropicalis as well. To my knowledge, not as much as 14,000 genome, but we now have 4,000 whole genomes fully sequenced on the server and allow for further study. So using the publicly available genome from Sender, I found that the mitochondrial genome for C. elegans is highly polymorphic. The mitochondrial genome on C. elegans is about 14 kb long, and I found about 1,300 polymorphism across the genome. So that's average 1 SNP per 10 base pair. And there's hundreds of missense mutations segregated in the species. And my population genetic analyses combined with site frequency spectrum suggest that these missense mutations are deleterious on average, as they are found at lower frequencies compared to synonymous mutations. We did include some of the sister species C. inopinnata in the original analyses, but these two species are very distantly related, so that kind of undermined the usage of this outgroup as to infer the evolutionary history of C. elegans. Regardless, we can largely classify the mitochondrial genome of C. elegans into 8 distinct phylogenetic groups. Next, I use principal component analysis on the nuclear genome and to see whether the mitochondrial genome and the nuclear genome co-evolved or not. And we kind of see a considerable concordance evolution between the mito and the nuclear in C. elegans population. So this suggests that exchanging mitochondrial DNA between individuals from different lineage should lead to the disruption of co-adapted mitonuclear complexes and reveal substantial episodic interactions. But how can we do that? It's now possible to exchange mitochondrial DNA between strains of C. elegans in one step without many back-crossing generation. We take advantage of this protein called GPR1 protein that regulates the chromosomal segregation in the first mitotic division of the C. elegans embryo. The overexpression of this protein leads to the premature segregation of the pronuclei, and this produces a viable offspring, which is chimeric. And then because C. elegans self, so the next generation, you would have a cybrid worm with a paternal nuclear genome paired with a maternally inherited mitochondria DNA. So taking advantage of this construct, I was able to generate a strain panel with unique mitonuclear genotypes, and I exchanged mitochondrial DNA between 18 different isolates from sender. And to capture the largest amount of genetic diversity, I chose 3 strains from each different 6 lineages that I showed earlier. And I was able to build, I think the largest to date, 325 unique mitonuclear genotype. I also reintroduced the mito back to the matched nuclear genome for control. So now we have the population, and to quickly assess how mitonuclear epistasis contribute to phenotype, we phenotyped just a subset of these strains, 6x6 panel, with 3 biological replicates per strains. We chose worm length as the phenotype of interest because it's a quick phenotype to score, and it's a proxy for development, which is energy constraint. We adopted a high throughput pipeline to phenotype worms across a variety of environments, including the lab environment at 20-degree, high temperature, 3 environments that are heavy metal that's proposed to be mitochondrially taxing, and paraquat, which is known for targeting the mitochondria. Basically, worms were grown in 48 hours and then score the length after that. This work would not have been accomplished without the help of the very talented post-bac student, Danny Klein. Here's the result. So in the for the control condition, so you can see the interaction plot here that it's represent the average worm length of all genotypes in the control condition. So each line represent a nuclear background, and each point represents a unique mitonuclear combination. You can clearly see a strong nuclear effect here, right? As some of the nuclear backgrounds grow smaller or bigger than the other. If we focus on the red nuclear here, sometimes the mitochondria alters the average worm length of that nuclear, suggests mitochondrial DNA has an effect on phenotype. But not every nuclear background responds to the same mitochondria the same way, so that indicates mitonuclear epistasis. We then quantify the broad sense heritability in these from this data, and all further petition the contribution of each genetic term to VG within each environment. As you can see here, the nuclear has the largest contribution to genetic variance, and mitochondrial and nuclear interactions also contribute substantially to genetic variance. Even though I showed you earlier in the interaction plot that mitochondria contribute to phenotypes, but on average, across all nuclear, there's no mitochondria that confer bigger or smaller worm size. So that's why you don't see any mitochondrial contribution in this plot. And I did that for everybody. As you can see here, again, mitonuclear interaction explain a significant proportion of genetic variation among the strains, ranging from 3% to 29%. Mitochondrial genome contribute to as much as 0.5% to genetic variance, and has the biggest effect on high temperature, which is consistent with the work in yeast and flies, and which suggests that the mitochondrial effect is most likely masked by the strong mitonuclear interactions. But for overall, the take-home for this experiment is that we show that mitonuclear interactions are pervasive across conditions. So let's go back to our original question. Do we see any evidence for mitonuclear coadaptation? Does a change in mitochondrial DNA between strains generate significant epistasis? So the answer to that, I'm analyzing 4 strains consisted of 2 matched mitonuclear combination and 2 mismatched mitonuclear combination. And I used 2-way ANOVA to assess the impact of nuclear, mito, and mitonuclear epistasis for each pairwise comparison. And I did this for 15 possible pairwise comparison across 6 conditions. And I'm just presenting a couple of examples here. In this interaction plot, you can clearly see a strong nuclear effect again and no mitochondrial effect. And a significant epistasis. What you can also see here is that the matched combo are larger than the mismatched combo in this particular condition and within this two pair. But that's not the case for the other two strains in Paraquat. In this case, the mismatched combo are larger than the matched combo. And in fact, this seems to be the trend for the Paraquat condition, where mismatch are much larger than the matched combo. So is there any evidence for mitonuclear co-adaptation at all? We reason that mitonuclear interactions are very sensitive to environment, and most likely selection for optimal interactions happen in ecologically relevant condition. And we're looking at these conditions in heavy metal and paraquat, which is not really informative. And also, worm length is not fitness. And if we really want to look at evidence for mitonuclear co-adaptation, we have to dig into the live history traits such as fertility, longevity, and pair with molecular phenotypes like ATP production and ROS generation and stuff. But I hope that convinced that with just a simple phenotype, we have been able to show the pervasiveness of mitonuclear interactions in C. elegans to phenotype. And this allow us, provide us a resource for further develop the model into an experimental evolution model where we can see how mitochondria constrain the evolution of a population. So stay tuned for future studies. And with that, I would like to thank my lab members and happy to take any questions. Yes.
Yeah, lovely work. I guess, so you were showing us the differences in the mean length. Did you see differences in variance also that you started looking across?
Great question. I have not, but I think introducing mitochondrial DNA into a nuclear background generally create a lot more variation in response. Yes.
So there's a lot of combinations that you measured, and if I understand correctly, please correct me if I'm wrong, there were certain combinations that really changed the response to heat, and there are others that did not.
Right.
Many others that did not.
Yes.
So why did you use the word pervasive? It's not pervasive.
But it's there.
It's there, but not pervasive.
Okay.
But it's important. I mean, there are some combinations that will affect the phenotype. And you want to focus again in that, understand the mechanism that underlies the importance for such combination to respond to stress. But there are others that did not. It is hard. Why pervasive?
Yes.
So when you were talking about all of these, like, basically mitochondrial nuclear combinations, are all of these like homoplasmic? And like, how well does your technique deal with like heteroplasmic variants and like stream transfer knowledge?
So that's the next question I want to ask to see what is the level of heteroplasmy in these hybrid worms. I don't have an answer for that yet. Stay tuned. Yes. Oh, that's it? Okay, sorry. Yeah. Yes.
Okay, hello everybody. My name's Aiden Pierce. I'm a third-year PhD student at UCL, and today I'm going to be talking to you guys about patterns of gene loss in endosymbionts at the origin of eukaryotes, or more specifically for you guys, how to make a mitochondria. Now, in order to kind of address this question, we need to address endosymbiosis at its most basic form. Now, endosymbiosis is just simply one organism living inside of another. Now, in this little guy that we have up here, this little aphid, inside of its gut, it's got specialized cells called bacteriocytes, and inside of these little bacteriocytes, you can see all these GFP-tagged Wolbachia. Now, these Wolbachia provide nutrients for the aphid, and they allow it to survive in nutrient-deficient environments. And in the same way, when you're living with an organism inside of your own membranes, in in the same way of you living with flatmates or living with a partner, you need to make compromises. Someone's got to take the bins out, someone's got to do cleaning, and you work together for that. Endosymbiosis requires biological compromises. And some of these biological compromises include streamlining of your metabolomics, your transcriptomics, and your genomic processes. For this talk, I'm just going to be focusing on the genomic processes that are going on. And there are the most clear transition that we can see from free-living bacteria to endosymbionts and then organelles of endosymbiotic origin are genome size. So whilst I could talk about things like the AT bias and stuff, I could talk about increased sequence rates. It's very clear that when you have something like a Wolbachia, it's got about a 1.5 megabase genome, which is considerable when we're considering endosymbionts, but it's still considerably smaller than free-living bacteria. And then you've got this axis where at the far end of it you have things like mitochondria. Now, how do we get such small genomes in mitochondria and endosymbionts versus free-living bacteria? Well, if you're a little endosymbiont, you can't recombine your DNA freely. In the same way that when we're walking around this conference, we're talking to people, we're saying hello, we're making eye contact, we're shaking hands, high-fiving, whatever. If I was to lock the door of this room, eat the key, and make sure no one can leave, because we've got a small population, we can't freely do that anymore. And so it makes it a lot more easy for mutations to enter the population. We can't fix our DNA. And so we get this rise in fixation of deleterious mutations. And these loss-of-function mutations in endosymbiont populations lead to deletions of certain genomic regions. Because if you're a mitochondria, if you're an organelle, if you're an endosymbiont, if you get a mutation which knocks out the function of a gene, then it's no really benefit to have that non-functional gene within your genome, and you're going to be outcompeted by things which can replicate faster with that gene that has already been eroded. And so we see these patterns of deletions within organelles and endosymbionts in general. Now, the interesting thing is that when you're an endosymbiont, the genes that you have not only have a fitness effect on yourself, but on the host that you're living in. And so if we're considering patterns of gene loss and this transition that we saw from free-living bacteria to endosymbiont to organelle, we need to consider that each gene that is being lost along this process has a fitness effect on us, the endosymbionts, and the room that we're in the host. So the question that I kind of want to make you all consider now is, considering these, how do we actually get an organelle? Are all endosymbionts along the same axis? Are we just waiting a matter of time for the genes to be lost, and so things like Wolbachia are eventually going to become an organelle? Or are they fundamentally different processes, such that the initial conditions are incredibly important and they will never get organelles from things like Wolbachia? Now, I've tricked you all. This is a theoretical biology talk, so I'm going to describe the model that I do this kind of computer simulations in. And the computer simulations that I have are relatively simple in the sense that we just have a number of hosts, and inside the hosts we have a certain number of endosymbionts, and then inside the endosymbionts we've got a certain number of genes. Now, because I'm only considering endosymbiont genome evolution, we are looking at how the endosymbiont genome determines the fitness of the endosymbiont and the host itself. And the way that we do that is by having this generation which deletions enter the endosymbiont population. We determine selection at the level of the host and at the level of the endosymbiont, and then we select using a Wright-Fisher process the fittest individuals that make it to the next generation. But then if we're only considering endosymbiont genome evolution, how does the endosymbiont genome determine fitness of the host and endosymbiont? And in order to look at that, as I've mentioned before, each gene has a different effect on the host and the fitness of the endosymbiont. So I'm going to be using this notation here where we're looking at the fitness effect W on the E, the endosymbiont, and H host. And we can divide the genome into different classes based on their fitness effects on both the host and endosymbiont. So in blue here, we have genes which are good for the endosymbiont and neutral for the host. We have in red genes which are neutral for the endosymbiont and good for the host. And then in black, we've got genes which are neutral for both the endosymbiont and the host. These black genes act as a meaningful control for drift in the simulations, just so we know what the baseline is. Now, if you're an endosymbiont, what do you actually care about in determining fitness in your own genome? Well, quite clearly, you care about the genes which are good for you and the number of deletions that you have in those genes. The more deletions that you have in these genes, the less fit you're going to be. Makes a lot of sense. You don't care about the genes which are only benefiting the host, and you don't care about the genes which are not benefiting anybody. So you can just freely accumulate deletions in these genes. Whereas, if you're a host, the endosymbiont population can lead to we're looking at the total endosymbiont population rather than just the individual endosymbiont itself. So in these situations, what we care is the products that are being produced across each loci. So we're looking across the endosymbiont population, and if you've got no mutations across the endosymbiont population at a locus, you're producing lots of product. This is good. If you've got a deletion, you're producing less product. This is less good. And so therefore, the fitness of the host is lower. Now, what we've done there is we've defined a very nice system for showing how multi-level selection, or how a gene which has an effect at both the endosymbiont and host level might influence and be affected by host and endosymbiont population dynamics. But to actually prove this case, what I've done is we've got a load of endosymbionts, the same number of endosymbionts as there are hosts in a population. Selection at the level of the endosymbiont host should be the same, and I've just let the endosymbiont genomes evolve over time. And what I'm hoping to show you here is that if you have genes with different functions at the host and endosymbiont level, they are retained at different rates once they hit this stable point. As I mentioned before, the black genes, you see pretty much the vertical line going down. That's drift in the population. It's incredibly fast, and it's not preserving anything. And then what we can see in the red and the blue genes is a very nice asymptotic curve where mutation selection balance is working at a level to preserve genes at a certain frequency. So what we can actually see here is even though population size at the host level and the endosymbiont level is the same, and selection coefficient is the same, we're seeing a differential retention of genes. This shows that there is a multi-level selection effect in determining how genes are differentially retained at the level of the endosymbiont and host. So, because we now know that multi-level selection is the thing that affects the number and the type and the function of genes which are being retained in endosymbionts, what we can ask now is if we're trying to think about how to get an organelle like a mitochondria, how function can relate to this. So in order to think about the functional capabilities and possibilities of an organelle, we need to increase the complexity of the genome. So now we're not just considering these blue, red, and black genes, we're also considering genes like this yellow gene here. Now this yellow gene is a selfish gene, which is good for the endosymbiont, but detrimental to the host. And we also have these altruistic genes here in green, which are detrimental to the endosymbiont, but good for the host. And then we've got these nice little friendly purple genes, which are good for everybody. So, considering this, what I wanted to do was consider a scenario at the origin of eukaryotes. So in order to kind of expand the kind of range that we were looking at, I wanted to run simulations with a certain number of endosymbionts in a certain number of host cells. So along the x-axis on each of these heat maps here, we have a certain number of endosymbionts, ranging from 1 to 100, and each of these is within either 1 or 100 hosts. So interestingly, what we can see here is a wide range of functional capabilities that are retained at the end of our simulations. If you have a very large number of endosymbionts in a very small number of cells, what we have is a parasitic-like action where you see these selfish genes being retained. And so what this tells us is that the selfish actions of an endosymbiont might be determined either by the gene. It's kind of a chicken and egg kind of thing. But if we're considering how to get a mitochondria or an organelle, what we're looking for are genes which are very beneficial to the host, potentially even at the detriment of the endosymbiont. And what's very nice is if we consider things like the biophysical restraints at the origin of eukaryotes, where you've got an Asgard archaea and an alpha-proteobacteria, which have a similar cell volume, you're going to have a very small number of endosymbionts per host cells. So if we look towards the left-hand axis on each of these plots, and if we look potentially at having a larger host population, if an environmental thing like the Great Oxidation event was kind of funneling this, like being this main selective pressure of evolution at this time, we can see that endosymbionts will retain genes even if they are detrimental to themselves, if they are very few in a very large number of host cells. So if we want to try and make something like a mitochondria, if we want to make an organelle in general, what we're finding is that the starting conditions are incredibly important in determining the function of the organelle that comes out of it. And so just to summarize here, if we have a very small number of endosymbionts in a very large number of hosts, we can get a mitochondrial-like state. If we have a very large number of endosymbionts in a small number of hosts, we can get a parasitic-like state. And so concluding that, I just want to say thank you very much. To everybody in the lab, you can clap, go for it. And any questions, I'd be happy to take. I have considered that, and it makes a very complex model. So I haven't shown it here. But you can imagine if you are freely living, having the time where you're not spent inside of a cell, the selection pressures change dramatically because you're not being provisioned by the kind of cozy environment of a host cell. So when you are free living bacteria, you're going to be retaining a lot more genes for things like flagellar motility, like dealing with like viruses and so on and so forth. So you would assume that if you had free living stages in a life cycle of an endosymbiont, you're going to retain a wider gene repertoire. And that further kind of solidifies the idea that you're not going to become an organelle if you have those free living stages. So that's actually going to be what my next kind of project is going to be about. So one of the limitations of this model is because I'm only considering endosymbiont genomes, one of the obviously the key things about becoming an organelle is transferring your DNA to the nucleus and having that kind of access back to the organelle. In those situations, in my next model, I want to have a host genome so that you can look at transfer to the host. And so you can see the difference that affects the endosymbiont population when you have a copy of a gene going to the host and how that affects the dynamics in between. So ask me in about a year, and I'll give you an incredibly detailed answer. Cool.
Thank you for allowing me to speak today. I'm going to talk to you about the unique mitochondrial inheritance and the recombination in mussels and its effect on fertilization. So to start off, I'm assuming that the majority of you agree with me that mussels are delicious. And if you don't think that, then I'm hoping that at least by the end of the talk, you'll think they're interesting. So mussels are extremely important for the environment. They're classified as ecosystem engineers, and that's because they are one of the biggest habitat formation animals and species within marine environments all across the world. Not only are they ecologically important, they're also economically important. In Australia, mussels make up one of the largest parts of the aquaculture system, especially in Victoria, where they are the largest, most abundant shellfish species farmed. In Australia, they are located all across the southern coast. We have a couple of different species. It's very complicated. It's been a nightmare throughout my PhD about what species we have. In Victoria and Tasmania, we do primarily have our own native species, which is Mytilus punulatus. Then in Western Australia and South Australia, we have the introduced Gallop provincialis. There's been multiple introductions of this from all over the world, and we do have a hybridization problem in Australia. They hybridize with each other, they hybridize with the native species, it's all over the place. But most of the time we do have the native species with Mediterranean and Atlantic Gallop provincialis. So, as you're all aware, well, I'm assuming you're aware because you're in a mitochondrial symposium, it's usually maternally inherited. However, in mussels specifically and a couple of species of clams, they actually have mitochondria from both maternal and paternal inheritance. And then this is called doubly uniparental inheritance. So the way this works is that you have a male and a female mussel. They spawn. The eggs contain the F or the female type mitochondria, and then the sperm contains the M type. Sperm meets egg, the normal occurs, and then the egg ends up, or the embryo ends up with both the M type and the F type mitochondria. After about 24 hours, however, the M type eggs will retain the M type mitochondria, but then the females will selectively eliminate or severely suppress the M type mitochondria for them to end up becoming a female mussel. Now, because they have two mitochondrial genomes, they have the chance of recombining just like nuclear genes do. There are quite a lot of pros and cons in recombination, specifically in nuclear recombination. I mean, the biggest thing that is that it creates a lot of variability and it fixes deleterious mutations. But there has also been evidence of disadvantageous gene combinations because of recombination, and that they can actually cause mutations that can lead to certain diseases such as cancer. So what happens within these muscle species is that you have the M-type mitochondria that then recombines with the female mitochondria and then slowly replaces the M-type throughout the system. What is also known is that this recombined M-type will eventually become a new M-type and then replace all of the M-types within the population. So this recombination is known to occur within the control region of the mitochondria. The control region, if you don't know, that it is extremely important for replication of mitochondria. But there's also some evidence for the COX-3 gene or the CO3 gene, but majority of publications do say it's in the control region. The control region of the male is actually shorter than the female, and then when it recombines, they stick together. So during recombination, the male control region is retained, and then that's inserted into all the female coding genes. So you end up with male control region with female coding genes for the rest of the mitochondria. I wanted to see is, first off, do we actually have this recombination within our native species? And is this recombination occurring in the same place as all of the other previous research in the Northern Hemisphere? So I did this wonderful program called RPD, which is Recombination Detection Program, and we end up with this fun graph. So the blue line at the top is the F genome, so this is just like a control genome that I got off NCBI. And then we've got the M-type genome, which is the purple, and then the yellow is my suspected recombined genome. When you end up with this overlapping region, that signifies a recombination event, and this aligned with the control region, which was just at the beginning of my sequence, which confirms that we do actually have the recombination within the control region, which is the same as the Northern Hemisphere studies. Interestingly, out of everything that I found, I did find about 35% prevalence of this recombination event, which is higher than most other studies, but I will get to that in a little bit. I used these recombination-specific primers, which is the standard PCR, so I could detect how much recombination was occurring. And when I sequenced them, I sequenced just the plain normal F-types as well, and I found that we had both southern and northern descent of these F-type genomes. However, with my recombined males, they only occurred in females that had southern hemisphere F-types. So does this mean that our hybrids aren't necessarily compatible with this recombination, or is it just more likely to occur in our native southern hemisphere species? But the first thing I wanted to test was, does it actually have a fitness effect, and is it stopping reproduction within our species? So previous research looking at this recombination fitness effect is severely lacking. Pretty much the only thing that's been found is that the M-type is evolving more rapidly than the F-type, which I believe was mentioned earlier, and that the recombined sperm actually has a faster swim speed than the normal M-type sperm. So I hypothesized that this mitochondrial genotype will actually influence the fertilization. We just have no idea if it was going to be positive or negative, because you think faster sperm might hit egg faster, but then because sperm in mussels swims in a circular motion, because it's going faster, it could just completely miss the egg altogether. Could just be like, go off in the distance. So I spent couple of months during the middle of COVID spawning mussels, which what else are you going to do? So I spawned them, and then I did my PCR determination just to see if they are recombined or not. I counted all my sperm and eggs just to make sure it remained consistent. I did use a homologous mixture of eggs just so I didn't have any sperm competition problems. And then I combined them to eventually, hopefully, fertilize them, and then I counted for fertilization. So first thing I looked for was these polar bodies, which is the first sign of fertilization. And then I looked for dividing cells. They aren't your usual dividing cells. You're probably used to mammalian ones, which are nice and even. Mussels do weird things. This is about the third stage of recombination, fertilization, and it occurs about an hour and a half in. So what did I find? Nothing. So first thing was that just between positive recombination and no recombination, there was absolutely no difference between the two. There was a slight positive trend towards having higher fertilization with recombination, but it wasn't actually significant. So I decided to split polar bodies from dividing cells just to see if it was occurring faster, like there was a faster rate. And once again, there was no significant difference. So, but that increasing trend was still happening. So potentially with more recombination happening, we could eventually have a better fertilization rate than the original M-type, but so far there is no difference. So just to summarise, the recombination definitely occurs in Australia. This is the first time that anyone's actually ever looked for it. And we do have this 35% prevalence. So it's important to note that almost every other publication that has been studied on this, there's been between 5 and 30% recombination. So we have a 35%, which is increasing. It could be that we just have higher recombination rates, but it could also be linked to the fact that the recombined mitochondria does eventually overtake the normal M-type. So we could be in that crossover event where the recombined genotype is becoming more preferable, so it is slowly getting rid of the normal M-type. And eventually it'll become the M-type again. There was no significant difference in fertilization success. However, we don't know that if it changes over time. So this is just the initial fertilization. We don't know if growing them up into adulthood will impact it. So what I would love to do next is I'll find the prevalence in all of those hybrid species to see if I can actually find recombination in the hybrids. And I want to do the post-fertilization test to see if it is actually impacted into adulthood. I'll also go further into the genome analysis because they do have female coding genes and we don't know what an impact of having female genes in a male mussel does. That's one of the next steps. I would just like to thank everyone who helped with my project, specifically the Queenscliff Marine Facility and Hatchery who helped me with all my spawning trials, which is the beautiful green thing in the middle, and I'm happy to take any questions. Hi.
Thanks for the talk, super interesting. I couldn't help but notice that you have about half the N for your positive group than you did for the negative. Can you comment on why you have such a lower number?
Yeah, so it pretty much aligns with having the 35% of recombination compared to the non-recombination. So there was a third of my samples that had the recombined mitochondrial DNA. I did do a power analysis to make sure that I had enough sample size to actually get significant results because it was such a difference. So it was mainly just to align with the fact that I had 35% recombination.